guesses? What do you think biochemistry is coming from? What was that?
I don't know, maybe organic.
Optimists would say, yeah, it's coming from organic chemistry. In my opinion, it's all organic chemistry. It is strictly applied organic chemistry. So one of the things that you're going to be responsible for are knowing all of the functional groups from organic chemistry. Some of them you won't see, like nitro groups, pretty minimal, aren't all that important. Some of the other ones listed up here, in fact, pretty much everything in that top, we won't evaluate to a great detail in biochemistry. So pretty much all of those we can ignore. Everything down below shows up fairly frequently, at least in differing forms. Okay? Why that differentiation? What's different about top versus bottom? I would argue that none of these really have stereochemistry. We see wedges and dashes, but that's only supposed to represent that three-dimensional structure. We don't really see stereochemistry in these drawings. Stereochemistry is massively important when we look at biochemistry. Not as difficult as in organic, because in organic it was which one do you have? In biochem, it's we have this one. It will always be this one. Yeah. Yeah, you're looking at a lot of oxygens and nitrogens. What's special about oxygen and nitrogen? They're very electronegative, which means they generate a lot more electron polarities. That polarity is what we take advantage of, or what biology took advantage of as far as generating the different interactions or new bonds within proteins, DNA, or RNA. Okay, so there are other functional groups. Your textbook has a nice figure. Table 1.1. I didn't have, and I still don't have access yet to the images from the textbook. Ideally, that's the one I want to put up here, because there are a bunch of other functional groups that show up. For instance, when you look at ATP synthase, we're looking at ATP. Okay, what did A, T, and P stand for? Okay, sorry. Adenosine triphosphate. That phosphate portion, okay, is ultimately probably the more important part. Okay, why is the A or the P of our ATP really important? What happens to ATP? Yeah, say it louder. You got it. Becomes ADP. So adenosine diphosphate, we lose one of those phosphates. In the process, what else happens? Plus a bunch of energy. Whoops. So why is that going to be particularly important? Well, the bond that broke to go from ATP to ADP is a phosphodiester bond, right? Or a phosphate anhydride bond. That energy stored in that bond is what biological molecules take advantage of to transfer and make and break other bonds. Right? So we need to be aware of what bonds are breaking when we're doing these energy transitions so that we can then transfer that energy to something else. Okay? So you need to be aware of all of those individual functional groups and always tie it back. Otherwise, all you end up seeing is what we've got written here. ATP goes to ADP plus P plus energy. Ultimately just says, I change a bunch of letters to make some new letters, and I know nothing about the chemistry. Okay? What we're trying to get at least through a chemistry version of biochemistry, is looking at those actual bonds and evaluating the chemistry that's actually occurring. We're going a lot back to organic chemistry. Okay? So, review those functional groups. I will ask questions about them, particularly from that figure. Where does biochemistry come from? Really what we're looking at is a couple common themes and different branches of it. So one of the things that we find within biochemistry is that we use the same general biomolecules. Okay? Those same biomolecules function and react with each other as we would predict from organic chemistry. Those biomolecules are your amino acids to make proteins, your nucleotides to make your DNA and RNA. I think I got that one right. Okay. So all life as we know it is manipulating some form of these biomolecules. So if we can get some understanding of how the biomolecules work on their own, we can then start to look at special cases, look at different proteins, look at different forms of life, and kind of propagate from there. Okay? They'll all use some form of energy. We already talked about ATP. So when we're looking at biochemistry, not only do we have to look at the chemistry of the bonds changing, but we also have to look at the energy changes. Okay? Where is that energy going? How is that energy transfer? Is it something that's allowed to happen on its own? Do I have to add something else to it? Okay? Really, to evaluate that, we're looking at physical chemistry. Okay? I don't think anybody's had physical chemistry yet, right? I think you've really, or you'll see it a little bit in physics. Okay? That physical chemistry aspect is usually the bane of all chemists that have to go beyond organic chemistry, because it ends up getting a lot more difficult. You end up quantifying things, you get into calculus and all that other fun stuff. We'll limit the exposure to that physical chemistry side a little bit, but we'll still have to go through some of that mathematics, and based on that quiz, it looks like you guys could probably all run circles around me when it comes to mathematics. So hopefully we won't have any math issues there. Okay. That was, I guess, a pleasant surprise. And then we could also look, when we're looking at biochemistry, kind of the beginning. Okay, where did all life come from in general? Which you could argue is looking at philosophy or even religion. Okay? How do we take some inanimate organic objects and bring them together, and now all of a sudden they become animate, self-replicating thought processes? Okay? Of these kind of three sections, really the only ones that we have any real understanding of are the first two. That whole philosophy of how we're bringing life together, how this leap actually happened. We can come up with theories, and there are some experiments to discuss it. But for the most part, it's a very dynamic field, to put it politely, I guess. Lots of debate happening within that. So we can start with the philosophy. We're not going to look at it a whole lot. And it is one of the potential lecture topics at the end. So if you guys wanted to look at what research has been done in trying to look at the origins of life and how you can bring those, or how those different biomolecules were formed, you can go ahead and do a presentation on that. Okay? One of the original kind of thoughts is what things were here to begin with. And so in general, most everybody agrees that these small molecules were available to us. None of these are animate objects. They're all inanimate, and depending on who you talk to, you might actually also hear the argument that they're all non-organic. Okay? It's a little bit weird, it depends on your definition. Okay? Some of the definitions for organic chemistry is anything with carbon in it. So we do have 3 with carbon. Okay? I don't quite accept that as an organic molecule. But it depends on who you talk to. Okay? So somehow or another, we had all of these molecules, and then we also had some kind of energy source, be it from solar radiation, be it thermal radiation from the Earth. All of these were somehow then mixed, and somehow we got life allegedly to come from it. So how did that actually all come together? So one of the seminal research projects that actually happened was looking at the Miller-Urey experiment, which in theory you all heard about. Yes? Okay, I see one head shaking yes. So at least one person's heard of it. Okay, so what they went through and did is combined a few of these different gases, and then they built this kind of enclosed system. Okay? And they then fired in a bunch of, what did they have, electrical energy. They do have heat. I thought they had UV in there too. Maybe not. At least not in that figure. It's not showing that. So they fired in a whole bunch of different sources of energy that would be something that we would predict to find in kind of our primordial Earth. And they let it run. And they just let it do its thing on its own in its nice little closed environment, because they wanted to see what would happen. Right? If we had all of these fundamental pieces, could we actually build life out of this pot? Right? Well, they didn't make any life. But what they did end up finding is that they were able to make other small molecules. Those small molecules were legitimate precursors to things that we use. Right? I don't think they got as far as amino acids or all of the amino acids, but they got some hydrogen cyanide, okay, which is a very reactive species. They got some formaldehyde, and I think they got some small amino acids. Okay? I don't think they got nucleotides out of this one. Other versions of the experiment, I think, have generated nucleotides. I don't think the original one did. And I can't remember if they're still running it just to see what happens. One of the big issues with this is that when we look at our origins, we're going back millions of years. Okay? And it probably took millions of years to build these larger molecules. And what we're attempting to do is give it a month, a year, and hope to magically we get life coming out of that right away. It's not going to happen. We don't have that time scale involved in it. Okay? So that time reference is a really important aspect that we really just can't correct for. And we could make adjustments, say, well, maybe let's assume with that time scale we make some amino acids. So now let's start with a whole bunch of amino acids, which may have been thousands of years of time to generate all of those. Now what happens? Can we get larger molecules out of that? And so that time scale becomes a very big issue. Yeah.
Is that what you said? They're probably still running it?
They might still be running it. I'm not positive, but it makes sense for them to still be running the experiment.
When did they do it?
Good question. That's what I was going to guess, in the 50s. I should know a whole lot more about it because they were based out of UC San Diego, which is where I did my undergrad. So I should know a whole lot more about it. They had little displays and everything all over campus for them. But I don't, because it's biochemistry. I like organic better. Okay? So it's kind of an interesting experiment. We've gone back and changed some of those conditions. There's been debates over whether their energy source was valid. I've also heard debates over whether or not they were allowed to use some of those molecules, like water. Okay? Should we have had liquid water in that system? Okay? So pretty much anything that we're going to generate is going to generate some contention along with it, and we're trying to ultimately just see how we could potentially work forwards. Okay? So it's kind of neat in that respect, but beyond that, we haven't found that kind of missing link that says this is the instant when we switched from inanimate to animate, or what we've defined as animate at least. Okay? And not to distract from it, their original focus was looking at proteins, because one of the original theories was that proteins were the source of life. Proteins were the first things that developed because they can catalyze other reactions and kind of propagate forward. As we've continued to do research on these, we've found that RNA can also self-catalyze. So it might very well have been RNA that started everything off and not proteins. So you're getting different branches from this on what started our origins. All right? So when we try and do that shift from the philosophy to the chemistry, really what we're saying is we start with these building blocks. We've got our amino acids, okay, we've got our nucleotides, we have our sugars. How do we then propagate out into the larger structures that we see in everyday life? Okay, and that's really where the biochemistry class is going to pick up. So when we look at all of those, ultimately, everything in life is a polymer. Okay? So biochemistry is massively about polymer chemistry. How many of you took any science on polymer chemistry? How many of you had a lecture on polymer chemistry? Depending on your professor, you'll see maybe one, maybe two lectures on polymer chemistry. Right? It is a really important field. It's very rarely discussed, largely because it's a really big field, so it needs its own class to really kind of get through it. Biochemistry is ultimately a study of polymers with very specific monomers, and we have different versions of those polymers. Proteins, okay, are a polymer. So what we're looking at is composing longer structures or bigger structures from repeating units. What are the repeating units in proteins? Amino acids. When we go up to the oligosaccharides or the polysaccharides, we're adding a whole bunch of sugars together. Okay? So each of the monomer units in that case are different forms of sugars. All right? And then we could jump all the way up to DNA and RNA, and what's happening with our monomer repeating units in there? You're looking at the nucleotides being added in differing patterns. What controls where those amino acids or nucleotides or which sugars go into this? That is dependent largely on the information that we've got in front of us. And again, that's where chemistry picks up and we kind of ignore the philosophy end of it. Okay, we're really just saying, we know that to make this sugar, this enzyme does it, and it'll put them together in this correct order every single time. Okay? And so why it does that is a whole separate can of worms that we usually tend to ignore. Okay? At least to a certain extent. When we jump from chemistry to biology, and this is where I'm going to be the absolute weakest, okay, what we're looking at is now that we've got all these kind of self-replicating particles, how are they organized on the larger extent? And that's where we build up to cells. And then so we could start to look at the different forms of life. And yeah, at this point I'm almost just reading off the slides. We're looking at prokaryotes and eukaryotes, kind of doing that division, and then what those differences are. And some of that comes back to the origins of life, and so you can hear different theories about where the eukaryotes come from versus the prokaryotes. Our prokaryotes are the, make sure I got that right, yeah, are the simplest forms of life. So you're ultimately looking at all of our self-replicating pieces are now trapped inside a membrane. And that's about it. Okay? So no real organization behind it. We've just kind of condensed where all of that chemistry is occurring. When we look at our eukaryotes, they've now expanded, okay, or divided those individual jobs and said, okay, I want all of the DNA to be replicated over here. I want all of the RNA to be replicated over here. I want all of the proteins to be done over here. I want all of the energy generated and pushed through the system over here. Okay? So our eukaryotes have kind of divided up most of those jobs. Well, to do that division, you're ultimately looking at what we would refer to as organelles. Okay? So we tend to see a lot more organelles showing up within the eukaryotes. Okay? So that's where you're seeing the nucleus. According to the textbook, and I don't know Greek, apparently eukaryotes is true sections or something like that. True pieces is the Greek translation for eukaryotes. So what we're looking at is, again, those kind of organelles showing up. So we got our nucleus, the mitochondria, okay, yet another organelle. What happens in the mitochondria?
Energy.
That's where we're looking at our energy sources. Our nucleus is our DNA. I don't know why I keep looking at the board. I can cheat and look at my iPad. The endoplasmic reticulum, which I typed out once and then decided that was too much effort. Okay. It could have been worse. I could have put in an extra C. What happened in the endoplasmic reticulum? You're looking at your protein synthesis. Chloroplasts, what's happening there? You're looking at photosynthesis, so another form of energy production, okay, if we're looking at a photosynthetic cell. Okay? At least in my opinion, our photosynthesis seems to happen in specialized sections, both in prokaryotes and eukaryotes. Okay? The chloroplast is a lot more structured, but we do have chromatophores. Okay? So that photosynthetic, that energy harvesting and conversion does show up in both in almost kind of a centralized location. It's kind of an interesting process, at least in my opinion, okay? Which means we could very well have been seeing some kind of symbiosis between an energy production and trying to do our DNA translations and all that fun stuff. Okay? So we end up combining different forms of life. Okay? Other random general facts, because I think they show up on the sapling homework. Your prokaryotes are looking about 2 billion years older. Okay? Why would we expect them to be older than our eukaryotes? They're simpler. Okay? It makes more sense for the simplest thing to start up first than it does for the more complex. Your prokaryotes are unicellular. Again, that goes back to the simplicity of them. They can't really propagate out and do larger groups. To a certain extent, we have found prokaryotes and small groups can actually kind of work together in kind of a symbiotic fashion, but nowhere near to the level of complexity that we get out of the eukaryotes, like a liver, right, or our lungs. We don't get that large compartmentalization that we get in eukaryotes. Okay. We already talked about our multicellular and all that fun stuff. Questions there? Please, God, no. Like I said, I swear, this is the biology aspect, hands down. You guys will run circles for sure around anything I can come out at you with. Okay, so I'll try and hit the big topics, but that's about it. And this one gets even worse as far as I'm concerned. Okay, how do we classify individual groups? What?
I said, describe that slide.
Yeah. Well, this is showing all of life divided up into our 5 sections and 2 main groups where we've got our eukaryotes and our prokaryotes. How's that work? How relevant this is for biochemistry? Because biochemistry does overlap with biology, it's going to show up in your first chapter, and they're going to kind of discuss it a little bit just to kind of give credit to the biologists. And then we will swiftly, as quickly as I can, shift away from it into the chemistry aspect. Okay? So you will probably need to review that Chapter 1 biology aspects, because you'll see questions in the sapling homework. I probably won't ever ask you a question on the biology aspects, because if I really have problems answering it, I don't expect you to. Okay? What I'm more concerned about is, can you apply some of this knowledge? And to me, this isn't really application. Okay? The next or last section of Chapter 1 is looking at energy. Okay, remember, we've got the physical chemistry aspect of biochemistry. So what we're going to be concerned about is, how do those reactions, okay, now that we know we have a reaction, what's the energetics behind them? Can they actually work? Do they work? Okay, do they have the energy to process forwards? Okay. If we're looking at life, okay, what can we say about all of the reactions occurring? Okay, are they all spontaneous? They will have to be spontaneous reactions. It's the only way they're going to propagate forwards. Okay. How does life make a reaction that we would typically, if we just look at the bonds changing, okay, that might very well be non-spontaneous? How does biology or biochemistry allow that reaction to occur?
It changes shape.
It changes the overall reaction dynamics. So we end up breaking or making a bunch of other bonds, and in that sequence, the overall process pushes it to being a spontaneous reaction. So we can look at a single step, and yes, we could have non-spontaneous reactions, but what biology is going to do is link that single step with several other reactions, so that the overall result is that we end up with a spontaneous reaction. Okay? Otherwise, things aren't going to propagate forwards. Okay? We're always kind of pushing towards that lower energy state. Okay? Fun little random fact. If we look at the conversion of organic materials to CO2, so we take a look at gasoline, and we combust it with oxygen, we produce what? Water and CO2, right? Okay. That reaction, spontaneous or non-spontaneous? Spontaneous. Okay. Once you provide that initial energy spark, boom, it continues on. Okay. We get that exothermic reaction. Our products are lower in energy than our reactants. That's with gasoline. Gasoline, organic molecule. What are we made up of? Organic molecules. Is the combustion of our bodies, CO2 and water, a spontaneous reaction? Yes. It is absolutely a spontaneous reaction. Then why on earth do we exist? Biochemistry has built in a massive energy barrier, okay, to prevent that reaction from happening quickly. Just because it's spontaneous doesn't mean it's fast. So when we think about energetics, we have to be very careful to separate these two concepts. We have thermodynamics and we have kinetics. The thermodynamics looks at the raw energy change between reactants and products. It does not care how much energy needs to get put in. We're only looking at the difference at the bottom. I color matched that awfully. Okay. I copied and pasted it from an old slide. I still can't believe I didn't go back and fix it. Okay. Our kinetics is looking at that energy barrier, our activation energy. Okay. So when we go through and look at our individual biochemical reactions, we will be looking at energy differences, which is ultimately looking at the thermodynamics. Okay. We will also look at kinetics. The kinetics is really going to show up in our second section when we're looking at enzymes and how we can then rank those enzymes in catalysis and enzyme kinetics and Michaelis-Menten equations and all that fun stuff. Okay? That's where all the math will come back in, all sorts of plotting, graphs, and other fun things to make it look easier. Okay? So what we need to do is be able to separate these two concepts. Not only do we have to separate them, we have to know what goes into it to make it up. Okay? So in biochemistry, we'll look at metabolism. Okay? Metabolism is how we transfer that energy around. And we can divide that into two separate sections. Remember I said we always want to go lower in energy. But to go lower in energy, we might have to go higher in energy somewhere else. Okay? So what we are looking at is a balance between adding energy into our system and removing that energy. Instead of saying adding energy and removing energy, which to me mathematically makes sense to just refer to it as that, this is, keep ripping on biologists, I'm sorry, because I know some of you are biologists. Instead of saying adding energy and removing energy, what do we go with? Catabolism and anabolism, that one, anabolism, C and A. Okay. So what we're looking at is increasing the energy and decreasing. Okay, we're looking at those exo versus endothermic reactions. And we end up dividing that topic into those separate sections. So in the self-presentations that you'll do at the end of the semester, okay, energetics is one of those big topics. It's also one of the reasons I'm a little bit nervous about not strictly lecturing on that, because that is an important aspect of biochemistry. And I'm really kind of putting that on you guys to be able to present and show that information well. Okay, so if you pick a topic that's dealing with energetics, there's a lot of cool things there. I do like it. One of the things that you're going to really have to address is, first off, which form of metabolism are you looking at? Or maybe you're looking at them both. Okay? And then what energetic differences are going on? What pieces are releasing that energy or increasing our energy? And what versions of it are taking that energy away? Where is that energy going? Okay? Or how is that energy being transferred? Okay? Questions about that? Okay, now I think I'm going to give you guys a chance, if I remember correctly. Yeah, our thermodynamic basics. We will come back to these topics. I intentionally did not put any symbols up here because I want you to kind of think about both the definitions and what symbols we end up using for these. Okay? So feel free to talk to the people around you, see what you can come up with. Hopefully it's a review. I know some of you with some of the potentially weaker chemistry backgrounds might not have seen this. Hopefully you have. In any case, we will end up defining it in just a little bit here. Okay? So please talk to the people around you. I'm going to pause the lecture. So you tell me, am I right? Did I miss something? Hi. We'll be talking about science a little bit later, yes.
I thought I remembered entropy being something with like environmental and external factors and stuff as well, or is that?
Your systems and surroundings?
So when you're looking at system versus surroundings, that's ultimately how you would calculate the difference. When we're looking at any of these, I guess technically the difference is a bad thing to be putting in there right now. Enthalpy is the symbol of H. Okay? Entropy is a symbol of S. Gibbs free energy is, we give the symbol of G. The delta that we put in there is the change. That change is the change between our system and surroundings. That's how we calculate that difference. Yeah, it doesn't really define it. No. Okay. So within this, probably the only one that really stands out as drastically different is our entropy change. In most cases, that entropy change is so small that it typically gets dropped from your calculations and everybody tends to not think about it. Really the only time that entropy really shows up and really gets discussed is in 152. Let me go through and say this is strictly what we're talking about when we look at energy. Really what we're looking at our entropy is our increase in motion, okay, or disorder. Disorder is the standard way to describe it. I usually prefer motion. If we were going to look at a reaction, okay, in fact, actually, let's take a look at ATP to our ADP plus our phosphate. In the course of that reaction, what happened to the entropy? Okay, so we could look at a system and its surroundings, or we can look at a before and after. Okay. Our entropy increased. So the sign on entropy in that reaction would be positive. Okay. And it increased because what happened to the number of particles? That increased, which is ultimately an increase in motion. In one case, we only have one particle that can move. In the second case, or in our products, which I guess I should step the other direction, has two particles that can now move. We've increased the possible motion within the system. Okay? Disorder is another way to look at it, and it works very well in this kind of 1 to 2 system. Kind of up to you how you want to address that. I'll try and always refer to it as motion, but that might be a bit mean because everybody's probably heard of it as disorder. Right? Those of you that had 152, how did they define entropy? Disorder or motion? Disorder. Yeah, I keep doing that. Sorry.
Sorry about that.
It's usually defined as disorder. Okay. Those of you that had me, had better remember I talked about it as motion. Okay. Just because I think it's more sensical for all of the definitions. When we look at our enthalpy, it's really a change in energy, but typically we monitor that by the change in heat. And that heat is ultimately being sourced from where? Is it a C in sourced? Good. Doesn't look like an N in the C. The U looks like an N, maybe. Sourced from our bonds breaking. Forming sounds a lot better than what I was going to write. So we'll stick with forming. I was going to say making. So bonds breaking and forming is ultimately where we're looking at our enthalpy. Okay? Those differences in energy between what bonds do we break, what bonds did we make, will help us kind of generate that. Okay? The very last thing, our Gibbs free energy, is ultimately how do all of these energy contributions come together to represent the overall energy dynamic. Okay? So that's our total energy. That's when we're looking at how the enthalpy interacts with our entropy. Okay? Our entropy is largely dependent on temperature. Why would it be dependent on temperature? What happens when we increase the temperature? Everything moves more. So our entropy and our temperature are very strongly correlated. So our temperature has to show up in with that. Okay? So we're looking at that change in total energy. If we went back to our diagram, where do all these symbols show up? Oh, crap, it's already on there. Well, that defeated that purpose. Well, let's go ahead and talk about, hey, go back one more. Okay, so we could go through, and where it says delta G at the bottom, we can say that's delta H minus T delta S. Okay. The reason we want to go back to this is when we talked about our overall reactions, what did we say was a good reaction? Where our products are lower in energy or higher in energy than our reactants? Lower in energy. Everything through chemistry is striving for the lowest energy possible. Okay? Lowest charge, lower energy. Okay? So we always want our products to be lower in energy. What does that mean the sign is on our delta G? Man, you said that too fast. Favorable delta G, we want it to be negative. And that's because we're going to subtract our product energy, final minus initial, or product minus our reactants. We'll always end up with a negative if our products have a lower energy. Okay? Let's propagate that further out. Okay? What sign would we expect for delta H?
Negative.
Okay? Why do you say negative?
The sign with delta S.
Okay. What we want to do to come up with favorable signs for each of these is try and limit our possibilities. We said delta G needs to be negative, okay, based on our overall assumptions here. We're now going to look at delta H. Let's remove delta S from our system. Let's say it is so small that the only thing we have to worry about is the energy coming from the bonds making and breaking, forming and breaking. Okay? So what sign does delta H need to be to give us a favorable delta G? It would also have to be negative. Okay? So we would hope our delta H is also negative. What happens with our delta S? Okay? So we could go through and do all of our mathematic manipulations and fun stuff like that. Or what we could try and do is say, okay, again, simplify. Delta H is now super small. Okay? So we'll make delta H 0. This term needs to be negative to be a favorable situation. Well, what happens if delta S was negative? The negatives cancel, we end up with a positive value. Okay, so what we're looking at for a favorable reaction is our delta S needs to be positive, so we get an overall delta G being negative.
That's how you get awesome products, right?
Which?
What? A positive delta G.
Awesome products?
Yeah, like
Awesome.
TNT.
Yes. Yeah. And that's one of the things that biology will end up going through and doing, is if we take all the energy found in a glucose molecule and we try to combust it, we would probably blow up the car. Okay? We would literally probably combust just within a couple molecules of glucose. Biology goes through and takes each of those individual bonds within the glucose and very carefully breaks them. And in the process of that, can now take that energy and put it somewhere else. Because if we completely broke all of those bonds instantaneously, we're going to have a massive release of energy, and that's going to be a bit problematic. Okay? So biology is all about trying to manipulate our H's, our delta H, our delta S, in such a way that we can release that energy very slowly and put it directly where we want it to be. Okay? One of those sources could very well be making ATP. Okay? So if we went back and looked at our overall reaction, the one we were talking about earlier, ATP to ADP plus our phosphate, and technically actually plus our energy, what's our delta G for this reaction? There's a big hint. The energy shows up on the product side, meaning that the energy of the molecule of the structures had to be lower than our reactants, and that's why that energy was released, which means the sign for our delta G for this reaction is negative, because it was a spontaneous reaction to release that energy. What's the sign on our delta H? It's also negative. That one's a little bit, hopefully, more intuitive if we think back to what our delta H was. It's that change in energy. If energy's on the product side, it's negative. If energy's on the reactant side, it's a positive delta H. We just talked about our delta S. Our delta S for this reaction was also positive. Okay, so this reaction is extremely spontaneous. Okay, it wants to go immediately. Or I should remove the immediate statement, because that involves time. Okay? ATP was our power source for all life, so we need ATP. Okay? How can we make ATP? If we try to reverse this reaction, plus our phosphate, plus our energy out here, that says energy, goes to ATP, that reaction is now non-spontaneous. It's a positive. ATP is our source of all energy. Okay? So if we're going to look at an overall, our life in general, are we really just saying that we hope we form ATP and then we can immediately use it? Okay? Or do we have some way to store that energy? We do have a way to store it, and we control that release. How can we make ATP later on? Well, we have to get an energy source from somewhere else. Okay? And that can be the absorption of light from the sun. That could also be the breaking of glucose molecules, okay, to go through and generate that energy. So what we end up doing in biology is linking these very non-spontaneous reactions with something else to make it spontaneous, so that we can actually sequence the thing all together. How to make sense? Okay. Where are we? Oh, look at that. I forgot. That was drawn much nicer there. So when we're looking at our phosphate group, you may or may not remember that from organic chemistry. That is the structure for your phosphate. I'm not going to attempt to draw the ADP or ATP for that matter. We'll save that for actually making images. I'm not going to hand draw that because it's too complex for me to draw quickly. So what we want to be aware of is how do these things shift around and form. When we go through and take a look at this reaction, what we're doing is linking this oxygen, okay, with AD let's do this so I can abbreviate. AMP, which stands for adenosine monophosphate. If I now put on another bond out to a phosphate, what do I have? Okay, ADP. I cheated. Okay, so there's ADP. Okay. If we now want to connect this to another phosphate, we have to somehow get this oxygen to link up with this molecule. Where's that oxygen going to link up? Why is it linked to the P and not to the oxygen that's floating out there? Right. If we were going to draw out our ATP, so now we'll again cheat. We'll do a DP. Oh, I think I have to draw it all the way out. Shucks. Our monophosphate. Here's our new phosphate.
You can't have a negative oxygen bonded to a regular.
Okay. Our phosphate are linked through an oxygen. Where did that other oxygen go? Water. Okay. Why did the oxygen link to the phosphorus and not to the other oxygen?
Too many electrons.
We could look at an electron-electron repulsion. The charge on our oxygen, negative. The charge on the other oxygen, negative. We're going to get a charge repulsion. They won't interact. Okay. Where do we have something that could interact with that negative oxygen? We need something positive. What can you tell me about that central phosphorus? Why is it positive? Because of all of the electronegative oxygens around it. Okay, so when you look at your individual reactions, look at how the reaction changes and say, okay, what had to happen? Why did that linkage occur? Okay, and that's going to be kind of the chemistry aspect of this, and that's going to be the part that really I'm going to be able to contribute to this class. Because beyond that, it's just, here's a new sequence of reactions, memorize the enzymes, the reagents, and all the bonds that changed. Okay? Which is a perfectly valid way to teach the class, don't get me wrong. Okay? That does absolutely happen, particularly if we looked at biochemistry, not biochemistry. I know, because my girlfriend at the time took biochemistry through the biology department, and I took it through the chemistry department, and we compared. It was like, what the hell are you learning? It's an entirely different subject. It was very bizarre. Other words that are going to show up, exothermic or exergonic. Not positive why we decided to switch that to exergonic. I still have to look that up. Or endothermic versus endergonic. What are those referring to? Energy released. Okay, it can be our delta H or our delta, well, it's officially our delta H. Okay. Our exo or xer are looking at delta H's that are negative. It's a release of energy, so we had to lose that energy. The mathematical sign for a loss, negative. That's what that sign means, by the way, loss. Okay. You're laughing at that, but that's a pretty big leap. When we originally look at the mathematics behind it, we just say, oh, it's negative. That means it's exothermic. Okay. Well, why? Because energy was released. That negative means energy shows up in the products. Mathematically, it's there, and sometimes we link that kind of what does it physically mean versus what it mathematically means, and we lose that connection. The endothermic versus endergonic, we're looking at a delta H that is positive. Okay? There's an equals in there. Okay? This reaction, what would we expect? catabolism or C or A? Okay, the reaction that's shown here. Okay, so let's erase everything so we can clean it up a little bit. Where's energy in this reaction? In the reactants, so we could add our energy into this. All right.
Is the H plus the energy?
No. Yeah. An organic chemist would actually have the structure drawn out here. And if you drew out that structure, you'll notice that you're missing a hydrogen to balance the water. Okay, to run that reaction, you need an extra H+. Which, if you go back to ATP synthase and how it actually forms ATP, one of the things that it needs is H+ passing through the enzyme, okay, to cause the rotation and force it together. We do need that H+. All right. I already forgot. What was the difference between our catabolism and
Metabolism is breaking down.
So think back to our energies, though. Okay, we're concerned about our energy. Our catabolism is breaking what? Breaking bonds. What does that do with our energy? I'm pretty sure I cheated and actually looked at it when I wrote the slides.
I just think of like anabolic steroids.
Right.
Well, it builds muscle, but to build that muscle, what do you have to do? You're going to have to get rid of that energy. That energy has to go somewhere. Okay. So if I actually wrote that correctly, our catabolism is an increase. Oh.
That's a pretty picture.
And I'm probably going to have to check that again for our catabolism. Current slide. Okay. In fact, I'm going to check that and get back to you guys. You guys were all positive it was anabolism.
Anabolism.
I was close that time, right?
Cannibalism, but take off the C, anabolism.
Anabolism. Okay. There we go. We'll see if that sticks.
We're going to make a biologist out of you,
Yeah. That would disappoint me greatly. But I'll accept it. Okay. So what was the verdict? We're going through and making bonds.
Anabolism.
So we're calling that
Building up.
as anabolism because we're putting those bonds together. Okay? Which meant that we were storing that energy. There's where the energy came in. Okay? And that's where our positives and minuses came through. The catabolism was the release of that energy. That's where we're getting that positive. Okay? That is officially pretty much everything in Chapter 1. The Chapter 1 homework is available on Sapling. I think they're all going to be roughly about 20 questions. If you actually have to draw stuff, I might reduce the number on the homework assignments. If it's just like matching, then you'll probably see more questions. The homework is Sapling Learning. It's online. There's in the syllabus, it mentions where to get it, and the syllabus is on there. If you go to saplinglearning.com and go through and sign up.
Is there going to be a homework
I'm going to mention that too.
each chapter?
Yes.
And when's that due?
Never.
When it comes to homework, for better or worse, I'm pretty lenient on due dates, particularly on a class that's just getting set up. All right. So if you go to Sapling Learning, you can buy it through them. Please don't buy it through the bookstore. It is $27 online. If you go to the bookstore, it's $37. And you're spending that extra $10 for the storage space of a tiny little slip of paper with a little scratch off. Right? I know scratch offs are fun, but I don't think they're worth $10. So please be aware of that. I want to get, because we've got the time to do it, because right, we're done at 9:30.
40.
40? Oh wow, we got a lot. Oh man, I need a lot more slides.
9:30.
Shoot.
9:25.
Yeah.
That was in organic, right? Was it required energy?
Yes, because energy was on the reactant side. Yes. Chapter 2 starts to look at the importance of water as an individual solvent, and they go on for 10, 15 pages about water, which I think very succinctly is summarized right here, right, as far as what's going on. And I think this applies better to everything that we'll be doing in biochemistry. Okay? It's also, for those of you that have had organic from me, it's also everything that happens in organic chemistry. Okay? It all comes back to ultimately this slide and manipulating it. Okay? So water ends up being a really special solvent in general. Okay? And your textbook goes, it's hydrogen bonding, and then it goes on for 15 pages about hydrogen bonding. Okay? The question becomes is, well, why is that important? Okay? Why do we care that water is such a special solvent? So we could look at a couple ways to approach this. We could look at the polarity of it, so we could actually start to look at the chemistry of it. We could also reverse engineer this. How important is water to life? Okay. Pretty much all of our life, okay, as we know it, is currently based around water as our general solvent. Okay? So water plays an extremely important role in pretty much everything. Okay? So it is going to be incredibly important. The question then becomes is, well, why is it so important? Okay, when we went back and looked at the functional groups, I know that was way, way long ago, okay, in these slides. And I had a whole bunch at the top, and I said you pretty much don't have to worry about those, but then the bottom I said were all important. Okay? What was the trend in those bottom that were important? Why were those important? We had electronegative atoms in them. We had oxygen and nitrogen. Those were really what drove the chemistry. Okay? Those functional groups end up being a lot more reactive. Why are they more reactive? Because they're electronegative, generating partial charges. That imbalance of charge now polarizes one end, a negative end, that could potentially go out and react with something positive, so we can generate a new bond. Or that positive end could now react with something negative. Okay, so what we're looking at is that imbalance between those charges and how that imbalance translates into what we can observe. Okay, and there are some simple observations that we can make. The big ones being our phase transitions, melting points and boiling points. Okay, those are all dependent on the individual molecules that make them up. And it's really a balance in between these two on trying to decide what's happening with those melting points and boiling points. I know most of you have not seen this version or this image before. That's because I made it. So let's go through and kind of interpret it. If we were going to take a look at two molecules, relatively the same mass, yeah. If we took a look at water and methane. Okay. And we were to look at, say, our phase transitions. Which one has the highest melting point? Which one has the highest melting point? Water. Which one has the highest boiling point? Water. Okay, well, that was a dumb question. You guys already know all those things. Okay, well, why? Why does water have a higher melting point? If you go back and evaluate the intermolecular forces, the attraction between a water molecule and another water molecule, that attraction is much stronger in water than it is between two methane molecules. Why? Okay. Hydrogen bonding is what we call it. Absolutely right. Okay. But why is that such a strong attractive force? Okay. Make it less scientific. You're right. And polarity is a big term that shows up, and it's also one of the terms I dislike. Most of my research was on polarity, and depending on who you talk to, it's a different definition. So polarity is usually a bad thing to make reference to. Positives and negatives. When we take a look at the water molecule, we are looking at an oxygen bound to a hydrogen. Okay. Why is that important? Well, oxygen, second most electronegative element, is taking all of the electrons in that bond and ultimately holding on to them. Whoa. Which means oxygen just stole an electron from hydrogen, so it becomes negative. So we get this massive partial negative charge. What happened to the hydrogen? It just lost an electron, so it becomes a massive partial positive. What happens with our methane? our carbon and hydrogen bond. The difference in electronegativity is almost identical. So the electrons are equally shared, which means what charge builds on the carbon? None. What charge builds on the hydrogen? None. There is no difference in polarity, or no polarity association with it, no dipole, which means we don't see a strong interaction between that molecule and another molecule. That's because we don't have a high energy state. That high energy state is those charges. If we don't have that charge, we don't have an interaction. So water is really special because that's pretty much as polar as we could possibly get, okay? And still allow for a relatively stable molecule. Okay? Because if we push to something, say, like HF, okay, that is still, it's technically a weak acid, but it's a lot more acidic than water. Okay? The difference in electronegativity is enough that that hydrogen can come off relatively quickly. So that molecule becomes very, very reactive, whereas in comparison to water. Whereas water still has that partial charge, so it does have some of that reactivity, but it's stable enough that it doesn't have to react. Okay? So it can allow for a lot of those potential interactions without completely destroying everything, okay? Melting bones. Okay? So why does this chart become important with that? Well, if you're given a molecule, in theory, hopefully, you should be able to classify the bond, right? So when we looked at the examples down here, you notice I simplified it. Instead of looking at CH4, I just looked at CH. What type of bond is there between a carbon and a hydrogen? It's a covalent, you're clearly trained by me. Non-polar covalent, okay? It's typically how it gets referred to. I just stick with covalent. Again, laziness, okay? If I have a covalent bond, what are the potential forces that that molecule can exert? Well, do we have partial charges? No. So it doesn't want to interact with another molecule. The forces that we get are London dispersion, very, very weak intermolecular forces. Okay? What happens when we move to water? What type of bond do we have there? A polar covalent bond. Okay? That can then translate over technically to a dipole-dipole. You said it was hydrogen bonding. Absolutely right. Okay? But I would prefer that you stick with dipole-dipole. Once you've established you have a dipole-dipole force, then you can evaluate. Is there hydrogen bonding? How do we know there's hydrogen bonding? Okay, this becomes important. You will see it in your sapling homework. You got to be careful with it.
Hydrogens are attracted to electrons?
What's that?
Hydrogens are attracted to electrons?
Close.
Electronegativity of the oxygen attracted to the hydrogen.
Okay, so that's very, very narrow. That does generate hydrogen bonding. But if I went through and asked you NH3, does that have hydrogen bonding? Yeah, there's no oxygen though. So your definition, while it worked for water, doesn't work for NH3. Oxygen, nitrogen, and fluorine. So to get hydrogen bonding, our extra definition is we have to have dipole-dipole first, then we can evaluate. Do we have a hydrogen bound to oxygen? Why did I write hydrogen? This is what I meant. Hydrogen, oxygen, nitrogen, or fluorine. Or said another way, like a colleague once suggested, we need hydrogen bound to phone. We need to have some phone. Right. Right, at least you guys find it entertaining. Good. Right? So we have to have that connection. Okay, so water gives us hydrogen bonding. So let's take a look at a couple examples, because I know these will show up. Whoa. Let's take a look at oxygen, hydrogen. So there's our first molecule. Sorry, that hydrogen looks a little messy. And here's our second molecule. Okay, those aren't molecules. Let me fix it so I don't get that complaint. What's that interaction? it's a trickier one. So, to determine what the interaction is, particularly because we're referencing hydrogen bonding, let's go through our rules. Before we can get to hydrogen bonding, what do we have to have? Dipole-dipole. Do we have a dipole generated in this molecule? Yes. Difference in electronegativity between our oxygen and our hydrogen. Do we have a dipole in this molecule? There's a large difference in electronegativity between the oxygen and the carbon. What interaction do we have between those two molecules? Sorry, we have a dipole-dipole. Now that we have a dipole-dipole, to move to the hydrogen bonding definition, what do we need to have? A hydrogen bound to oxygen. Bond, yeah. Do we have a hydrogen bound to oxygen, nitrogen, or fluorine? Yes. The interaction we have in there is hydrogen bonding. Okay. Seems a bit odd because if we took a look, now let's simplify this out or change this around and let's make it the exact same molecule. What interaction do we have in here? Now it's just dipole-dipole. Okay? We don't have that hydrogen bound to an oxygen, nitrogen, or fluorine. What happens if I now ask this interaction? This one's actually really tricky. So our far right one is absolutely dipole, right?
Yeah.
Do we have a dipole between carbon and hydrogen?
No.
No. Do we have a dipole-dipole force? No. At best, what we're looking at is a London dispersion dipole force. Okay? So it should be stronger than our standard London dispersion force, but it's going to be much weaker than our dipole-dipole. Yes, we have a hydrogen in there, but that hydrogen is not bound to foam, which means we can't have a hydrogen bond. Okay? I would argue that that explanation is completely unnecessary because before you get to that definition, that extra step, what did we need to have? A dipole-dipole. We don't have that dipole-dipole. We can't even look at that where that hydrogen is located. Okay? Kind of makes sense? So you will run into that. So how does that then apply back to, all our intermolecular forces? Well, what we can do with our phase transitions is look at the strength of the force. The stronger the force, the stronger the attraction between molecules. What is that going to mean about the amount of energy I have to put in to break those molecules away from each other? It's going to have to go higher. Okay? So we can see our melting points and boiling points going up. What about solubility? Okay, what happens when we actually dissolve something? To get two species to mix. Okay, like dissolves like is the standard way to memorize it. Okay, I won't call anybody out, but I would adjust that. Like dissolves like is too vague. It's like what dissolves like what. Like forces dissolve like forces. So evaluate the two molecules you're looking at. Are the forces similar? If they're similar, we would expect them to mix. If they aren't similar, we don't expect them to mix, and they'd be insoluble. Okay, so you could go through and evaluate, just again, which of those forces do you have? It doesn't work perfectly, but it gets you pretty close. Okay? Why does it have to be similar forces? Okay, so that's picking dissimilar forces.
That's what I'm saying. If they weren't similar, they would, like.
Why do they not mix, though?
Repel each other.
Why do they repel each other? I would actually argue they aren't even repelling each other.
Because one's polar. They're more attached to each other than.
They're more attracted to the molecules that make them up than they are with the other one. So if I want two species to mix, if I've got hydrogen bonding in water and then London dispersion in the oil, for those two to mix, I have to have a balance in the energies. There's a lot of energy in that hydrogen bond neutralizing that charge. For me to throw oil in there, water now has to interact with the oil. And it has to interact at least as strong as the water did with itself. If it's not as strong of an interaction, what happens to the charges on that water molecule? They go up higher relative to when it was dissolved in water. The higher the charge, the higher the energy. High energy is bad. So what does water do? It completely removes itself from the oil because it can't balance out that charge. Okay? So that's what's happening on that physical level when you look at solubility. You could push that all the way to reactivity, your acid-base chemistry. That's ultimately what's happening with those, exchanging hydrogen ions to make sure that you can balance out that charge. We are officially done. Okay, sorry. By a couple minutes.
Does anybody